In this video, we will conclude our discussion of the chemical methods of microbial control as we analyze the use of surfactants, heavy metals, aldehydes, gaseous agents, and enzymes as forms of microbial control. Surfactants include detergents and soaps, which act to reduce surface tension. Soaps are molecules that contain a hydrophobic portion composed of fatty acids on one end of the molecule and a charged component on the other end, much like the phospholipids we studied in Unit 1. The hydrophobic portion of the soap causes emulsification of the oils on the surface of the skin, meaning that it breaks it into tiny droplets. The debris and microbes trapped in the oils on the skin are carried away by water. So soaps do not act as antiseptics, but rather assist in the mechanical removal of microbes. You may hear them referred to as degerming agents. Detergents are agents that are more soluble in water than are soaps. The most widely used group are those known as the quaternary ammonium compounds, which, as their name suggests, contain an ammonium cation. These compounds can act as germicidal agents by interfering with the integrity of the cytoplasmic membrane. Quaternary ammonium compounds are effective against vegetative bacteria, fungi, and envelope viruses. They are ineffective against endospores, mycobacteria, and naked viruses. Some species of bacteria can also survive in quaternary ammonium compounds. In fact, Pseudomonas actively grows in the presence of these compounds. Quaternary ammonium compounds are colorless, odorless, tasteless, and non-toxic except at high concentrations. A disadvantage of these compounds is that the presence of organic materials does interfere with their action. Quaternary ammonium compounds have been used in mouthwashes, ophthalmic solutions, cosmetics, and have been used to sanitize floors, furniture, and equipment surfaces. Some common ones you may have encountered are benzalkonium chloride, also known as Zephyrin, and cetylpyridinium chloride, which is used in Cepacol mouthwash. Heavy metal ions have been used to control microbial growth. The metallic elements mercury, silver, gold, copper, arsenic, and zinc have all been used. These elements act to denature proteins but only serve as bacteriostatic and fungistatic agents. Use of compounds containing heavy metals poses several problems. One important problem is that many of these metals are toxic to humans even in small quantities. People may also exhibit allergies to them. Another problem is that many microbes develop resistance to these compounds, and these compounds are often inactivated in the presence of organic materials. Some examples of metal-containing compounds that have been used for medical purposes are 1% silver nitrate and thimerosal. 1% silver nitrate used to be placed on the eyes of newborns to prevent blindness caused by Neisseria gonorrhea. This treatment has been replaced with safer, more effective antimicrobials. Thimerosal, which contains mercury and is a metabolic poison, was used as a preservative in vaccines. Thimerosal has largely been replaced with safer alternatives and is only used in a few adult vaccines. Compounds containing a carbon-hydrogen-oxygen functional group on the terminal carbon are referred to as aldehydes. Two aldehydes that have been used as antimicrobials are formaldehyde and glutaraldehyde. These agents work by cross-linking the functional groups of amino acids in a protein, causing them to denature, and also inactivates nucleic acids. They are among the most effective antimicrobials. Formaldehyde is a gaseous agent that is typically dissolved in a 37% aqueous solution known as formalin. Formalin is an effective disinfectant that has been used in the preservation of biological specimens, embalming, and in the disinfection of medical equipment. Formaldehyde, however, is irritating to human tissues, has an unpleasant odor, is carcinogenic, is slow-acting and leaves a white residue. Glutaraldehyde is a liquid and has been proven to be more effective than formaldehyde. In addition, glutaraldehyde is less irritating, retains its potency even in the presence of organic matter, is non-corrosive, and does not damage plastics. Glutaraldehyde works as a disinfectant to effectively eliminate bacteria, fungi, and viruses within about 10 minutes. It is also a sterilant, but the materials to be sterilized must be exposed for 10 hours. Glutaraldehyde is accepted as a high-level germicide and a sterilant. It has been used to sterilize medical equipment such as respiratory therapy equipment, hemostats, and kidney dialysis equipment. Gaseous agents are microbicidal and sporicidal gases that are utilized in specialized chambers that look much like steam autoclaves. The gases act to kill microbes by cross-linking functional groups, causing proteins to denature and damage to the structure of DNA. These agents are highly penetrating and offer an alternative to the sterilization of materials that are damaged by heat, radiation, and liquid chemicals. Gaseous agents include ethylene oxide, propylene oxide, beta-propiolactone, and chlorine dioxide. Ethylene oxide is a commonly used agent. It is a colorless gas at room temperature, and it is highly explosive, can cause damage to the lungs, eyes, and mucous membranes, and is carcinogenic. Ethylene oxide, when used correctly, can act as a sporicide. Propylene oxide is a chemical similar to ethylene oxide and has the same mode of action, but is less toxic. Chlorine dioxide is a gaseous agent that is typically used to treat drinking water, wastewater, food processing equipment, and medical waste. Chlorine dioxide was the agent that was selected to decontaminate the Senate offices following the anthrax attack of 2001. These agents are frequently used in hospitals and dental offices to disinfect and sterilize delicate instruments, and have been used to sterilize foods. There are enzymes naturally produced by organisms that work to inhibit the growth of microbes. One excellent example is lysozyme. We briefly studied the use of lysozyme by bacteriophages as a means of entry through cell walls and as a mechanism to rupture the cell wall to allow the release of the virus. Humans also contain lysozyme within tears. Scientists are currently researching the potential for the use of other enzymes as antimicrobials for industry and medical applications. One important enzyme is one known as prionzyme, which acts to destroy prions and may be used to eliminate prions on medical equipment.